So we're gonna move on to our final speaker of the session, Hilde Nelissen from VIB at Ghent in Belgium. So floor is yours.
Hi, thanks everybody. I realize I'm the last thing that keeps you from lunch or from the weekend if you're in Europe like me. So I will try to move quite swiftly through this. So different from all the other presentations that you heard today, I will go into more targeted approach. So it's not about sequencing and looking whole genome wide, but actually really looking at a targeted approach. Very briefly, our team is interested in studying growth processes, so mainly in maize. And as you can see, these growth processes take place throughout the life cycle of maize. And a lot of these processes originate in the shoot-apical meristem, which is very much at the base here. And I show this because most of the images I show are about the maize shoot-apical meristem, and I just don't know if everybody is so familiar to look at this. So there is a dome of cells that are undifferentiated, and at the flanks of the shoot-apical meristem, we have primordia that emerge, which we call P1, which is the last emerged primordia, P2, and so on. So if we make such a transverse section, and I will show several of those, through the maize shoot-apical meristem, what we see is the pool of undifferentiated cells. We will see different stages of differentiating primordia. And here at the bottom, we often see the pit region where the stem later originates from. Okay, then I move into technologies. So the most data that we acquired are from the molecular cartography data set, but we've also been working with Cartana that now is acquired by 10x Genomics, and we worked with that in the past. And these days we're also exploring VisGen. But for simplicity, I will mainly show the molecular cartography data. And so the idea is actually that this resembles like an in situ hybridization, but that you do this with multiple genes simultaneously. And different from what Stefania just told you, you do this with a predefined panel set. So if you were to do this with in situ hybridizations, you're always working on different sections, even if it's the same tissue, and it is very difficult to allow the comparison. While if you use a targeted spatial transcriptomics approach, or even an untargeted, you can visualize transcripts simultaneously in the tissue context. So although I said I mainly show the data from Resolve Biosciences, there are many different providers for targeted spatial transcriptomics at the moment. All of them have slightly different chemistry, and it was not my intention to be comprehensive here, but just to show there is a list. It is also, over the past couple of years, it was a moving landscape, because there were providers that, there were new providers, there were providers that were gone. And so I think if you want to do this, it's best to check out what the status is at a given time. Now, a lot of these providers initiate these studies for clinical studies, or these technologies for clinical studies, and not all of them are also adjusted or willing to move into the plant world. And there are several reasons for this. Some of them only worked with fixed gene panels, for instance, for diagnostics, to detect markers for certain diseases. And there is also a lot of difference. Some work with cryosectioning, and you heard from Stefania that there is some trouble with that. Some work with embedded and fixed tissue, which I will refer to as FFPE, or some actually are a bit agnostic and use both. So although there are many differences, a general workflow is that in phase 1, you will select a gene panel. Because this is a targeted approach, this is more towards hypothesis testing, and not like Stefania says that this will really lead to new identification. So this is a very critical step, and I will go back to this. So this is something that you, of course, do yourself. You select the probes, but then often you go back to the provider that will synthesize your probes, often on their proprietary knowledge on how to do that. In the next step, you will prepare your tissue type, and that then will be hybridized with the probes. And so the section, fixation and sectioning we always did in-house, but the hybridization and the imaging we always did in a service mode. However, if you buy your own device, this is also something you could do in-house. And then there is part on downstream analysis, which I call here segmentation, visualization, and data mining. Although this is not the main topic of this workshop, I still want to go into this slightly because it will show you that whatever you want to reach with this will somehow feed back into your gene panel selection. For these analyses, there were also solutions provided by the different providers, and we heard some today in previous talks. I must only say that not all of them are always tailored for plants. Some of the providers also feel more comfortable if you would perform a pilot experiment. For instance, there is a provider that would like to check the RNA degradation in your tissue sections, which means that you will make the sections, you will send them to them, and they will do a hybridization only with one gene. But so your tissue section goes through the entire pipeline, and you get a readout of the RNA quality by using the housekeeping gene. Other providers would prefer, if they work for the first time with a new tissue or a new species, to do a validation pilot with a small probe set. So this again depends on the provider that you choose. So then I want to come back to the phase one and the design of this gene panel. And I really want to emphasize that this is crucial. Of course, this depends on your biological question. We also heard in single cell analysis that spatial transcriptomics is actually a very nice way to validate certain clusters and especially for unknown clusters types. And I think especially the targeted is quite good for this because you can really select top 10 genes of your clusters. However, I think it's even when you do this, I think it is still important to include sufficient markers of the different cell types, especially when they are known. For instance, if I now zoom back to the maize shoot typical meristem, we already had a lot of in situs in literature. And so it was quite easy for maize to pick about 20 genes that we could think of that covered all the tissue types there. If you forget to do this, then later on you can come in problems. For instance, when you want to do the clustering, I will briefly show it, but also Stefania showed that you can use the spatial data as well for clustering. And if you don't have certain cell types represented in your data set, then this will bias the clustering as well. Now, in our studies, we always used different cell type markers, as I said, but we also selected highly expressing genes, low expressors, genes with a broad expression profile and with a very specific expression profile. And this was again based on what was known in literature, for instance, on laser capture microdissection or single cell data. And we did this because in the beginning that not much was known how this technology was working in plants. And we thought maybe the very specific genes will not pop up in the right expression profile, or we will have troubles with highly expressed genes that mask maybe lower expressed genes. Now, in the technologies that we benchmarked so far, none of these issues were actually at stake. So we were able to detect both highly expressed, lowly expressed, specific, and more broad expressors. So there was no bias towards any of those. So towards the future, I don't know if you still need to take them along. I can just give you the idea what we took with that. If you are working in a less characterized species, because I said in maize we already have quite a lot of markers, then still try to work with homologs for the different cell types. So at least you would have some ID and some markers for the certain cell types. And another thing that we now know, or that we now start to realize when we are in the data mining step, that it might be good for normalization when you ever want to quantify, to also consider to take along some housekeeping genes. We didn't do this in the past because we were afraid that very highly expressing genes might mask other ones. And now, of course, in hindsight, when we look at data analysis, it might be a handy tool to have a normalizing gene present. So that's then all in your lab, that you design the probes or the gene panel that you want to work with. Typically, we design or we select a little bit more than the ones we can go with. In the case of molecular cartography, they have a limit of 100 genes for the moment. Other providers like VisGen can go up to 500 genes. And so then you give this list to the provider, and then they run their algorithms on that, and then it comes back with a list of probes for which they can or cannot design, or for the list of genes for which they can or cannot design good probes. In the case of molecular cartography, the genes need to be long enough, so it's impossible to look for small RNAs because they want to tile the probe, so they have a stretch of about 500 base pairs that is specific. And for the same reason, it's very difficult to work with, to discriminate with similar genes. So if you have genes which have close paralogs or part of gene families that are very conserved, then also they fail to make you specific probes against this. Once you have done this, then you can make your final selection, and the provider will start to synthesize your probe panel. During that time, you can start with fixating and sectioning your tissue. As I said, we worked with the maize typical meristem a few days after the fourth leaf appeared. We actually used very little optimization for a standard in situ hybridization protocol. So we used standard fixation, we embedded it in paraffin. One point that I want to make here is, and doesn't go for people that are very experienced in situ hybridizations, but something to take into account, again, also for the price of this type of analysis, is that you really need to make sure that the way you make your sections, because of course you can cut in different planes through your tissue. It depends on how you oriented it. And if you're slightly out of focus of your tissue, of course, that is the outcome you will get. And you do this pretty much blind. I mean, if you have experience in sectioning, you learn this, but this is something to take into account. Typically, we use sections that are 7 micrometers thick, but I also know 10 micrometers works. The only 2 small optimizations we made, and they are not always necessary, but they can be important for more difficult tissue than the maize typical meristem, is to include a baking step of 60 degrees, because we worked also with some other tissues with collaborators, and yeah, because this is not our data, I cannot show it here today, but I can tell you that then sometimes we had a adhesion of the tissue from the slide, and for this reason we included the baking step, because it doesn't seem to affect the protocol. We now apply it standardly, and because we ship the slides back to the provider, we also code it with slow fade. Then once the provider, and in our case this was Resolve, got our slides, they did the first imaging. So it doesn't look like this yet, because that is indeed with gene info on it, but they send us a scan of the entire slide, and then we are about to select certain tiles. And as you can see, the tiles are, dimensions are mentioned here. So you can select about 60 of those tiles. And so it's up to you then to choose on which tissue you want to focus. For instance, if you have a large maize seed, you only want to have the seed coat, you can go the circumference of the seed. In this case, you can take more of the pit tissue or more of the leaf tissue as you want. They then do the hybridization and the imaging, and afterwards they send you a report on false positives, and they also already look at transcripts which are actually below certain cutoffs. And what they also give you then is a visualization of the expression patterns. That's pretty much what you see here. Now, if you look carefully, you also see that there is a tissue image at the background, and that is actually something that you need if you really want to start to segment the individual cells afterwards. And here, for once, there was an advantage of working with plants, because typically what these companies do is do a DAPI stain, so you visualize the nuclei. But of course, you know that that is still not sufficient to determine the circumference of a cell, especially not in plants. And if you think about the meristem, here in the meristem region, cells are rather small, spherical, and dense. But if you look in these leaves, cells already start to expand, and they actually become already rectangular, turgor pressure is increasing, and so on. So to visualize also the cell wall, we included the chalcofluor white staining. And so now typically both stainings are done at Resolve Biosciences for plant tissue. And so once we have those two, we can start to segment the individual cells. I'm not going to go into detail because I realize this is already data processing, but it took us some time optimizing different programs that we have to use, and also quite a lot of manual curation. But in the end, we were able to segment all the cells, also the vascular cells, and we also got rid of the tiling effect. So that's what you can see here. And now we can then actually map the reads for which we got XY coordinates actually to very little ROIs, because every cell can now be considered as a region of interest. And so when we map now these cells, the transcripts to the individual cells, we can start to do qualitative and quantitative or quantitative analysis with this. I will briefly show you. It has been shown already. You can do clustering on the data, and you can then map the clusters back on the tissue context. For every cluster, for instance, for the blue one here, you can see which of the genes are actually mainly responsible for this cluster. But we can also look more quantitatively. Here on your right, you can see actually gradients, because we know the exact amount of counts per cell. We can really make this quantitative. And so you see in yellow, we see the cells that are highly expressing this gene. And in blue, we see the lowly expressing gene. In gray, there is no expression. So you see that here, this is not at 1. We see that there is expression in the meristematic cells, but that the expression goes down towards the positions where the leaves are attached. We can do this gene per gene. Here is another example, but we can also look at co-expression. So if we now take those two genes and we put them on top of each other in the same slide, then you can see that there is knotted expression in blue and in yellow, but then the cells that only express the other gene are in pinkish here. And so this really allows you to look from a gene-centric point of view, whether there is co-expression, but you can also click on clusters or on cells and really check which genes are co-expressed in certain cells. It also allows to reconstitute trajectories. So in this case, this is one that we selected ourselves. So we took a a path going from the tip of the meristem, which is actually position 0, and we took different cell files until we reached the tip of P2, which is the second primordium, and this would be here the tip of P2, and then you can plot, for instance, these are 5 genes along this path, and then you could see that indeed, when knotted 1 goes down, some differentiation markers start to go up, but we see that there are some other genes also playing a role. And so this trajectory then really corresponds to a trajectory that was done in the Scanlon lab using single cell data. So with this, I hope that I have shown that targeted spatial transcriptomics can be a great hypothesis tester. It can be used across species. I mainly showed now maize data, but if you have a limited amount of genome information, and if you can embed and section your tissue, you could actually do it on virtually any species. You don't need any transformation. So if you want to confirm the identity of certain clusters, and it's not so easy to make marker lines, you could use targeted spatial transcriptomics. You can use it to just map expression and look how where it is in the tissue. But if you really want, you can also go into quantitative and qualitative differences. I didn't have time to show it here, but we have data where we used overexpression line or mutants, and then you could actually see both quantitative and qualitative differences in expression profiles. And to come back on the thing that I said in the beginning, to achieve all of this, it is very crucial that you think what you want to get out of it. Do you want to go to the clustering? Do you want to go to the single cell level? Do you want to do the segmentation? Because all of that, I think, is crucial to determine the gene panel that you work with. And with this, I would like to thank the team. These are the 2 providers I showed data for. For VisGen, we're still working on that. I got some help from bioinformaticians and some funding from the VIB TechWatch to do this. And I would like to emphasize that you can always contact us if you have any questions.